A warm welcome to Sirnagen Therapeutics, and thank you for joining us today. At Sirnagen, we are addressing the limitations of RNAi therapeutics through our proprietary breakthrough platform, SAMiRNA. This technology enables us to develop innovative therapies across a broad range of diseases. Today, I would like to highlight the unique advantages of our platform and provide an overview of our key pipeline programs. Before we begin, I'd like to start with a brief disclaimer. The information presented here today is intended solely for this meeting and should not be regarded as investment advice or a solicitation of any kind. Some of the statements we will discuss may be forward-looking in nature. These reflect our current expectations, but are subject to risks and uncertainties that could cause actual results to differ materially. With that said, our goal today is to provide you with an overview of Sirnagen Therapeutics, our platform, and our development pipeline. Now, let me walk you through the innovation of our SAMiRNA platform. First, it avoids activation of innate immunity. Unlike lipid nanoparticle-based approaches, SAMiRNA eliminates the innate immune toxicities that are commonly observed. Second, it enables delivery across a broad range of tissues and cell types. Beyond the liver, SAMiRNA achieves effective delivery to inflamed tissues, adipogenic tissues, tumor site, and immune cells in the blood. Moreover, by attaching specific ligands, receptor-targeted delivery can be achieved. Third, it provides room temperature stability and long-term storage. Its unique single-molecule self-assembly protects RNA, eliminating the need for cold chain distribution. Finally, SAMiRNA enables efficient and scalable manufacturing. With one-step automated solid-phase synthesis and simple LC purification, production is both cost-effective and reliable. Its self-assembling nature removes the need for complex formulation steps, ensuring consistent quality at low cost. Altogether, these features position SAMiRNA as a differentiated next-generation RNA therapeutic platform that uniquely combines safety, delivery efficiency, stability, and cost-effectiveness. Now, let me briefly walk you through the core strengths of our SAMiRNA platform across 6 major therapeutic areas, representing a combined market opportunity expected to exceed $1.5 trillion. First, in the central nervous system. Via a single subcutaneous administration with a glut ligand, SAMiRNA can non-invasively cross the blood-brain barrier. Second, in oncology. SAMiRNA not only targets cancer cells, but also reprograms immune and stromal cells within the tumor microenvironment, thereby restoring anti-tumor immunity and remodeling the tumor microenvironment into a state more responsive to therapy. Third, in pulmonary diseases. Through inhalation, SAMiRNA is efficiently delivered to alveoli and vascular endothelium, demonstrating an excellent safety profile without acute or chronic toxicity. Fourth, in respiratory viral infections. Inhaled SAMiRNA maintains durable antiviral activity and is effective across diverse viral variants. Fifth, in dermatology. With simple topical application, SAMiRNA penetrates to the dermal papilla, achieving therapeutic effects without systemic exposure. Sixth, in immunological diseases. SAMiRNA efficiently reaches inflamed tissues and circulating immune cells, enabling broad applicability across immune-driven conditions. In conclusion, the SAMiRNA platform uniquely addresses critical unmet needs across central nervous system, oncology, respiratory, dermatology, and immunology. This positions SAMiRNA as a differentiated, next-generation RNA therapeutic solution and a compelling partner opportunity in the global market. Next, I would like to introduce SAMiRNA, our next-generation RNAi platform designed for innovative partnerships. First, safety. In human studies, SAMiRNA has demonstrated an excellent safety profile, showing no evidence of innate immune activation. The completion of our Phase 1b study is expected in the second quarter of 2026. Second, delivery. SAMiRNA can be administered via intravenous, subcutaneous, or intramuscular injection, enabling precise delivery to inflamed tissues and tumor sites beyond the liver through the EPR effect. It also distributes efficiently to immune cells. With inhalation, it reaches alveoli and endothelial cells, and with topical application, it penetrates into hair follicles down to the dermal papilla. Third, efficacy. In preclinical studies, SAMiRNA has shown meaningful results across fibrotic diseases such as IPF, CKD, and MASH, as well as in solid tumors and respiratory viral infections. These preclinical findings translate into clinical proof of mechanism, as we have confirmed mRNA knockdown within hair follicles, and our topical anti-hair revitalizing tonic has already been commercialized. Fourth, speed and scalability. We have established an integrated CMC system that covers candidate design, large-scale production, and quality control. This enables new drug candidates to be developed in less than 2 months, supporting rapid clinical entry, commercialization, and global licensing opportunities. In summary, these 4 strengths, safety, delivery, efficacy, and scalability, position SAMiRNA as a transformative RNAi platform and a compelling opportunity for global partnerships. We look forward to driving innovation together. Next, I would like to introduce our pipeline for innovative partnerships. This high-value pipeline is designed to redefine patient treatment paradigms. First, in oncology with SRN-001. SRN-001 remodels the tumor microenvironment to synergize with immunotherapy, chemotherapy, and radiotherapy. This differentiated mechanism positions SRN-001 as a unique complement to existing cancer therapies. We plan to initiate Phase 2A in the second half of 2026, with combination trials in pancreatic and head and neck cancers, together with immune checkpoint inhibitors or radiotherapy. Second, in fibrotic diseases. As a disease-modifying therapy, SRN-001 restored organ function in mouse models of pulmonary and renal fibrosis. Phase 2 is scheduled to begin in the fourth quarter of 2026. Third, in alopecia areata with SRN-XXX. We have already commercialized an androgenetic alopecia product based on our hair follicle mRNA knockdown technology. In ex vivo human scalp models, we demonstrated restoration of hair follicle immune privilege. An IND submission is planned for the end of 2026. Fourth, in influenza A and RSV. SAMiRNA has demonstrated robust and sustained antiviral efficacy, including against drug-resistant and emerging strains of H1N1, H3N2, and H5N1. We expect to submit an IND by late 2026. In summary, this pipeline roadmap underscores our ability to address high-value diseases with differentiated solutions. We look forward to partnerships that will accelerate the development of transformative therapies for patients worldwide. Next, let me briefly walk you through the innovative design principles of SAMiRNA. First, SAMiRNA is synthesized through a single streamlined process on a standard oligonucleotide synthesizer, which simplifies manufacturing and ensures consistent quality. Second, within a single molecule, we integrate a hydrophilic polymer and a hydrophobic polymer directly onto the RNAi strand. The hydrophilic polymer can also be conjugated with various ligands for tissue-specific delivery. Third, this integrated structure naturally self-assembles into RNA nanoparticles at a clinically relevant size of about 100 nanometers. Fourth, without requiring any additional formulation, these nanoparticles overcome the key limitations of conventional sRNA therapeutics. Together, these design elements establish SAMiRNA as a true next-generation RNAi platform. Importantly, SAMiRNA is protected by a strong intellectual property portfolio with more than 190 patents worldwide, covering composition, methods, and therapeutic applications. Now, let me briefly walk you through the safety profile of SAMiRNA. First, on innate immune stimulation. Compared with ConA and lipid nanoparticles, SAMiRNA kept TNF-alpha, MCP1, IFN-gamma, IL-12, and IL-6 at baseline levels, showing no innate immune activation. Next, from GLP-compliant preclinical toxicology studies. In general toxicity, the no observed adverse effect level exceeded 300 milligrams per kilogram in mice and 100 milligrams per kilogram in non-human primates. Importantly, no dose-limiting toxicity or major organ toxicity was observed. In genetic toxicology, SAMiRNA was negative in Ames in vitro cytogenetic and in vivo micronucleus assays, confirming genomic safety. In safety pharmacology, modified Irwin testing, respiratory function, and hERG assays revealed no significant effects. Finally, in cardiovascular telemetry studies, cynomolgus monkeys received doses up to 100 milligrams per kilogram, with no adverse findings. Altogether, this comprehensive GLP dataset confirms that SAMiRNA demonstrates an exceptionally low toxicity profile and a strong safety margin for clinical development. Now, let me share the human safety results of SAMiRNA from our Phase 1a trial. First, SRN-001 showed excellent safety and tolerability after a single 4-dose. At up to 10 times the anticipated therapeutic dose, no adverse events were observed. Second, tolerability was consistent across all dose groups, from 15 milligrams to 210 milligrams. Third, plasma levels were measurable at every dose, with AUC increasing proportionally. Fourth, even at the highest dose of 210 milligrams, no significant changes were seen in inflammatory biomarkers. In conclusion, Phase 1a results confirm that SRN-001 has outstanding safety, broad tolerability, and a predictable pharmacokinetic profile across a wide dosing range. These findings strongly validate the clinical development potential of the SAMiRNA platform. Now, let me introduce the delivery efficiency of SAMiRNA across tissues and immune cells, which clearly differentiates it from conventional RNAi platforms. First, in tumor-bearing models, both intravenous and subcutaneous administration enabled selective delivery to tumor tissues. Second, in mouse models of pulmonary fibrosis and chronic kidney disease, SAMiRNA precisely targeted inflamed tissues. Third, in preclinical studies, inhalation ensured effective delivery all the way to alveolar cells. Fourth, in human ex vivo studies, topical application alone achieved direct penetration into hair follicles. Fifth, using both human peripheral blood mononuclear cells and animal models, we observed strong uptake into immune cells. Taken together, these findings validate SAMiRNA's robust and versatile delivery profile across diverse tissues and immune populations, firmly establishing it as a true next-generation RNAi platform. Now, let me share the cellular-level evidence of SAMiRNA delivery efficiency. RNA interference is a natural process that silences genes by degrading specific mRNA molecules. Inside the cell, SAMiRNA binds to the RNA-induced silencing complex, precisely cleaving target mRNA and blocking protein production. In vitro studies show that SAMiRNA penetrates human synovial cells and effectively suppresses target gene expression. This precise delivery enables selective silencing of disease-causing genes, offering highly targeted treatments for cancer, viral infections, fibrotic diseases, and autoimmune disorders. Now, let me share the results of SAMiRNA accumulation in the endoplasmic reticulum, confirmed by storm imaging. In COS, 7 cells treated at 40 nanomolar, we observed strong fluorescent signals in the endoplasmic reticulum region at 4 hours, and these signals remained clearly visible even after 8 hours. The intensity of the signals, represented on a blue to yellow scale, confirms selective enrichment within the ER. Importantly, the endoplasmic reticulum is the central site of mRNA translation into proteins. Therefore, the high delivery to and sustained accumulation within the endoplasmic reticulum clearly demonstrate the unique effectiveness of SAMiRNA in reaching this critical site of action. Now, let me introduce the pharmacokinetic and pharmacodynamic profile of SAMiRNA. It demonstrated sustained efficacy for 1 month after a single 4 administration. First, the pharmacokinetic results. Following a single 5 milligrams per kilogram intravenous injection, SAMiRNA rapidly reached its maximum plasma concentration, and stable blood levels were maintained for 4 weeks. Second, the pharmacodynamic results. At the same dose, target gene knockdown was sustained for more than 4 weeks. This long-lasting efficacy is enabled by the excellent pharmacokinetic properties of SAMiRNA and its innovative, self-assembled nanoparticle structure. Now, let me briefly present the storage stability of SAMiRNA. First, SAMiRNA nanoparticles maintain physical and chemical stability for at least 12 months in aqueous solution at 22 degrees Celsius and 55% relative humidity. Second, even after 1 year at room temperature, there was no loss of delivery efficiency or pharmacological activity. This means SAMiRNA can be stored and distributed without the need for a cold chain, an important advantage over conventional RNAi therapeutics. Such stability not only reduces costs and logistical burdens from manufacturing through to patient administration, but also enables efficient supply in regions with limited infrastructure. Altogether, this stability profile positions SAMiRNA as a uniquely competitive RNAi platform for global market entry. Next, let me introduce the simple and scalable manufacturing process of SAMiRNA. Unlike conventional lipid nanoparticle formulations, which require multiple steps such as liposome assembly and lipid mixing, SAMiRNA is produced in just 4 streamlined steps on a standard oligonucleotide synthesizer. Strand synthesis, purification, self-assembly through annealing, and lyophilization. By eliminating additional liposome formulation, the process is greatly simplified. All steps are fully integrated into standardized synthesizers, allowing rapid process transitions and seamless scale-up. This streamlined approach enables cost-effective large-scale manufacturing with consistent quality. Most importantly, it strengthens global supply chain resilience and ensures reliable, long-term value creation for our partners. Unlike conventional antifibrotic therapies, SRN-001 simultaneously modulates immune responses and suppresses fibrosis. This means SRN-001 is not just a symptomatic treatment, but a novel therapeutic approach with the potential to fundamentally control the progression of fibrosis. Now, I will present the efficacy of SRN-001 in IPF mouse models. IPF is a progressive disease with very limited treatment options. Amphoregulin plays a central role in fibrosis by acting both upstream and downstream of TGF-beta to drive fibroblast differentiation. SRN-001 directly targets amphoregulin to effectively block fibrosis progression. We evaluated SRN-001 in 3 preclinical models: bleomycin-induced, TGF-beta overexpression, and silicon-induced fibrosis. Following intravenous administration, SRN-001 significantly reduced amphoregulin expression and consistently suppressed fibrotic markers, collagen deposition, and inflammatory signaling. Histological analyses confirmed clear reductions in inflammatory infiltration and collagen accumulation. Pulmonary function tests demonstrated clear improvements in lung resistance, elasticity, and compliance, indicating an overall enhancement of lung function. In conclusion, these findings highlight the strong anti-fibrotic efficacy of SRN-001 and support its potential as a new therapeutic approach for IPF. Despite its rising global prevalence, chronic kidney disease still lacks effective therapies. Now, I will present the efficacy of SRN-001 in preclinical chronic kidney disease models. We evaluated SRN-001 in 2 well-established models: unilateral ureteral obstruction and adenine-induced nephropathy. SRN-001 effectively suppressed amphoregulin, confirming precise target engagement. Key fibrosis markers were significantly reduced in both models. Histological analyses revealed marked decreases in inflammatory infiltration, tubular dilation, and fibrotic area. Inflammatory mediators were also notably downregulated. Importantly, in the adenine model, blood urea nitrogen and serum creatinine levels approached normalization, demonstrating functional renal recovery. Taken together, these findings position SRN-001 as a disease-modifying therapeutic candidate with strong and durable anti-fibrotic efficacy in chronic kidney disease. SRN-001 represents a novel approach to cancer therapy by reprogramming the tumor microenvironment, thereby substantially improving the effectiveness of immunotherapy and radiotherapy. I will now take you through the key preclinical evidence and mechanisms behind this effect. Next, I will explain how SRN-001 reprograms the tumor microenvironment. First, it inhibits tumor cell proliferation and promotes apoptosis. Second, it suppresses metastasis, limits invasion, and mitigates drug resistance, thereby improving overall therapeutic outcomes. Third, SRN-001 reverses fibroblast activation and alleviates immune suppression, enabling T cells to infiltrate the tumor core. Currently, immunotherapy achieves only a 20 to 30% response rate, largely because the tumor microenvironment blocks T cell entry and amphoregulin upregulates PD-L1. SRN-001 suppresses amphoregulin, reduces PD-L1 expression, and restores T cell-mediated anti-tumor immunity. Moreover, conventional therapies remodel the extracellular matrix and drive resistance, while radiotherapy elevates amphoregulin, fueling fibrosis, immune suppression, and metastasis. By targeting these pathways simultaneously, SRN-001 establishes powerful synergy with immunotherapy, chemotherapy, and radiotherapy, positioning it as a transformative therapeutic approach. Now, let me explain the clinical impact of amphoregulin expression. High amphoregulin expression is consistently associated with poor survival across multiple cancers, including head and neck, lung, and pancreatic cancer. These findings were statistically robust across all cohorts. Taken together, elevated amphoregulin predicts poor prognosis and highlights the critical need for therapeutic strategies directly targeting amphoregulin. Now, let me explain how SRN-001 reprograms the tumor microenvironment to exert potent anti-tumor efficacy. First, SRN-001 promotes infiltration and activation of CD4 positive and CD8 positive T cells. Second, it enhances M1 macrophage-mediated anti-tumor immunity. Third, it reduces fibrosis and immune suppression driven by M2 macrophages. Fourth, it alleviates TREG-mediated immunosuppression. Fifth, it relieves checkpoint inhibition by lowering PD-L1 expression and reducing PD-1-positive CD8 T cells. Sixth, it blocks tumor metabolic adaptation by suppressing lipid droplet accumulation. In conclusion, SRN-001 achieves comprehensive tumor microenvironment remodeling by restoring anti-tumor immunity, reducing fibrosis, and preventing tumor metabolic escape. In a head and neck cancer model, SRN-001 demonstrated strong synergy in combination with an anti-PD-L1 antibody. Altogether, the unique tumor microenvironment remodeling capability of SRN-001 provides differentiated clinical value across multiple tumor types. Now, let me explain the relationship between radiotherapy and amphoregulin. Radiotherapy is one of the main pillars of cancer treatment. Worldwide, more than half of cancer patients receive it, and in high-income countries the proportion exceeds 70%. Research by Andres Pivko showed that radiotherapy increases amphoregulin levels in both tumors and blood, and this rise is strongly linked to worse survival. This has been consistently observed in patient samples and animal models. This process is called the Badskopal effect. Radiotherapy drives amphoregulin upregulation, which reprograms the immune system and fuels metastasis, a vicious cycle. In solid tumor patients, higher post-radiotherapy amphoregulin levels strongly correlate with poor survival. Similarly, in non-small cell lung cancer patients, even when radiotherapy is combined with checkpoint inhibitors, elevated amphoregulin remains consistently associated with reduced survival. Altogether, these findings highlight the critical role of amphoregulin in undermining the effectiveness of radiotherapy. Now, let me present the relationship between radiotherapy and amphoregulin observed in animal models. In mouse models, radiation exposure led to a clear, dose-dependent increase in amphoregulin levels across tumors, serum, and lung tissue. We also observed that lung metastasis rose proportionally with radiation dose. Furthermore, higher post-radiotherapy amphoregulin levels correlated with greater tumor volume. These results clearly show that radiotherapy is not limited to localized effects. Through amphoregulin upregulation, it can also drive tumor growth and metastasis in preclinical models. Now, let me share how SRN-001 has the potential to revolutionize radiotherapy outcomes. In animal models, the combination of anti-amphoregulin with radiotherapy led to a marked reduction in both tumor volume and metastasis compared with radiotherapy alone. Moreover, when anti-amphoregulin was combined with radiotherapy and an immune checkpoint inhibitor, anti-CD47, the inhibitory effects were further enhanced, showing outstanding suppression of both tumor growth and metastasis. These results are particularly important in the context of the so-called Bad-Scopal effect. By directly targeting amphoregulin, SRN-001 reprograms this process, transforming radiotherapy from a treatment that can unintentionally fuel progression into one that suppresses metastasis and enhances therapeutic efficacy. Altogether, SRN-001 represents more than just an adjunct therapy. It offers a powerful combinational strategy to fundamentally boost the effectiveness of radiotherapy and immunotherapy. Now, let me introduce our SRNAi platform. SRNAi is a next-generation siRNA-based therapeutic platform for respiratory viruses. Unlike conventional antivirals, SRNAi delivers potent inhibition by directly silencing viral genes. Its flexible design also enables rapid adaptation against emerging and future threats, including so-called disease X. Let me now introduce the unique value proposition of the SRNAi antiviral platform. SRNAi is built on a novel RNAi-based strategy that delivers potent and durable antiviral efficacy, with the flexibility to cover both emerging and future variants. Importantly, no innate immune activation or cytotoxicity have been observed, confirming an excellent safety profile. Its inhaler-based delivery system ensures efficient targeting to the lungs, maximizing therapeutic impact. Preclinical studies demonstrated broad-spectrum antiviral activity in vitro and in vivo, with greater efficacy and improved safety compared to existing treatments. In a lethal challenge model, SRNAi achieved a 100% survival rate. Altogether, SRNAi represents a next-generation platform with strong potential to transform respiratory antiviral therapy. Now, let me introduce the SAMiRNA platform. SAMiRNA is a next-generation, commercially available solution for hair revitalization, powered by our proprietary SAMiRNA technology. SAMiRNA will set a new paradigm in global scalp and follicle care. SAMiRNA is a game-changer in hair revitalization. SAMiRNA is a topical formulation based on our SAMiRNA technology. After application to the scalp, a simple massage delivers it deep into the hair follicle. Once inside, SAMiRNA selectively suppresses follicular mRNA as associated with hair miniaturization. SAMiRNA ARI specifically inhibits androgen receptor mRNA, which is overexpressed in the sebaceous glands and dermal papilla cells of androgenetic alopecia patients. By targeting androgen receptor mRNA, SAMiRNA ARI reduces excessive sebum production and prevents dermal papilla cell apoptosis, offering a fundamentally new approach to restoring follicle function. Through this mechanism, SAMiRNA provides an innovative solution for restoring follicle function. Now, let me present the results of the ex vivo human scalp study. When SAMiRNA ARI was applied to human scalp tissue, it penetrated into the hair follicles and dermal papilla within 6 hours, with clear cellular uptake observed. Importantly, this penetration was maintained for at least 24 hours. As a result, androgen receptor protein expression within the follicles was significantly reduced by more than 50%, providing clear mechanistic evidence for delaying follicular miniaturization. Now, let me share the results from the clinical study conducted by Dermatest in Europe. First, regarding safety, SAMiRNA ARI received the highest possible 5-star rating, clearly confirming its safety for topical application. Second, regarding efficacy. Even with only once-weekly or bi-weekly application, we observed significant increases in both the median density of terminal hair and the number of anagen hairs. This means SAMiRNA ARI does more than slow follicular miniaturization. It restores follicle function and stimulates hair regeneration. Importantly, these benefits were consistently observed in both men and women, underscoring its broad clinical potential. Now, I will present the unique value of SAMiRNA ARI. SAMiRNA ARI has demonstrated innovation, safety, and efficacy across multiple clinical studies. Its strong efficacy in restoring follicle health addresses the limited effectiveness of existing treatments. It received the highest 5-star safety rating from German Dermatest, confirming excellent tolerability with no adverse effects. SAMiRNA ARI is suitable for both men and women. With a simple once-weekly topical application, it also delivers outstanding user convenience. In doing so, it effectively addresses critical unmet needs that current therapies fail to meet. Backed by robust scientific evidence and global patent protection, SAMiRNA ARI has also gained international recognition, winning the 2024 Best Hair Product Award at the Bologna Cosmoprof Awards, underscoring its broad clinical and commercial potential. Now, let me move to the final slide. SAMiRNA ARI is currently marketed in more than 70 countries, including Europe and the United States. Through continuous feedback from customers worldwide, strong and consistent evidence has been accumulated for both its efficacy and safety. Most encouragingly, not a single adverse event has been reported to date. SAMiRNA ARI is built on the scientifically validated SAMiRNA technology platform, which demonstrates that SAMiRNA is becoming a true game-changer in hair revitalization. Looking ahead, we envision partnerships with pharmaceutical companies that bring expertise across diverse therapeutic areas. Through such collaborations, we aim to leverage the SAMiRNA platform to accelerate the conquest of intractable diseases and, ultimately, to save the lives of patients in need. Thank you.